Here we are continuing our discussion of lipids and really shifting into just membranes. So we know that membranes are going to be a big part of the purpose of lipids, and so we're kind of going to focus on that from here on. So membranes, of course, what they separate is different polar aqueous liquid sections. So we might have the extracellular liquid out here, and then we have the cytosol in here, and that's separated by a membrane. Or we might have the cytosol and then the matrix of the mitochondria, which is the liquid phase inside it, again separated by a membrane, right? So they're very good at separating two liquid phases because the liquid can't really pass through unless you give it a hole or something like that. So lipids are not polymers. They are not covalently linked to each other, but they will self-associate, and that is just to avoid water, essentially. And depending on what the lipid looks like, it will form different structures. So if these individual units are more wedge-shaped, this is what it would look like if it's a free fatty acid. They tend to make little micelles where they kind of all point in, and you have like one point here, and this is a slice through a spherical one, and then all coming out here, and you have all the little polar head groups sticking into the water. So this is, again, what fatty acids would do. This is how sort of soaps work. Soaps are generally some variant, they're often modified, but a variant of a fatty acid, and they kind of form a micelle around little bits of dirt or oil or whatever. However, if they are about equal cylindrical, and this is what our membrane lipids look like, either the sphingolipids or the glycerophospholipids, they tend to form bilayers, and that's because you can picture all the little cylinders lining up, but then they can't really necessarily loop around like this. However, a bilayer has an obvious problem, which is the sides, right? These fatty acids don't really want to be touching the water either, so if it's big enough, what you often get is that this loops around and comes back around, and you get then a vesicle. Okay, so a vesicle is often what we say if it's a very small one, but really a cell is just a giant vesicle, right, with a very large aqueous cavity on the inside. And again, all of this is formed from non-covalent interactions to simply avoid water. So we have many different membranes in our cells, and the different membranes have different lipids. You're not responsible for knowing this, although a few of the really standout ones that we've talked about already might make sense. So the plasma membrane has a lot more cholesterol than anything else. Cholesterol, as we'll see in a minute, tends to sort of help structural formation, and the plasma membrane is the one that needs to be like the most structurally sound, right? Because it's really separating the cell from everything else. And we can separate the phosphatidylcholine and phosphatidylethanolamine are the two most common glycerophospholipids, but certainly if you connect those two together and picture all the blue as being glycerophospholipids, they do make up the majority of lipids in most membranes. Maybe it's a closer call in the plasma membrane, maybe almost like a third, and another third with the sphingolipids and the minor other things that are making up the rest of it. So again, these different membranes, it's totally fine. They can and do have different lipid makeups. Even the two monolayers usually have a different composition, so the inner and the outer of that bilayer. And we'll see in a minute that can happen because there really is not a lot of movement between them. This is of a red blood cell membrane, so it's the plasma membrane of that red blood cell. We can see that the inner monolayer is very different than the outer monolayer. The outer monolayer has a lot of phosphatidylcholine and a lot of sphingomyelin. We've already talked about how sphingolipids play a role in blood type and things like that, and that's why they tend to be on the outer monolayer and not nearly as much on the inner monolayer there. And so this is just a number of other, obviously, glycerophospholipids. These inositol ones, those are involved in signaling. So the inositol sugar tends to get, well, it's a sugar alcohol, it tends to get cleaved off and then be a signaling molecule inside the cell, which would therefore make sense as to why you primarily see it on the inner monolayer. We can even see differences sometimes in different portions of the same membrane, and these can't be maintained forever because they will start to sort of even out, at least one monolayer and the other. But because things are constantly moving around, so you might have a transport vesicle that sort of buds off of the trans-Golgi and then goes into the plasma membrane. That's going to transport some lipids from here into the plasma membrane, or vice versa. It could happen the other way around. You might have something come in from the plasma membrane and then bind and merge with another membrane inside. So you can have stretches where it's not as obviously drastically different, like here it changes, but you can actually have some differentiation even within one membrane like this. Another thing to talk about is that membranes, I think we tend to sometimes think about them as just being a relatively boring lipid bilayer, almost like a soap bubble or something, except made out of lipids. But that's really not the case. There are a lot of other things going on in the membranes, particularly proteins. So we'll talk more about these. So we haven't been talking about proteins for a while, but it's going to come back. So if we just make a simple lipid bilayer from the normal fatty acids, the normal lipids that would make that up, it's only about 3 nanometers wide. So about 3 nanometers from here to here. However, if you could measure the distance of what's considered the plasma membrane of a cell, you would actually get about 5 to 8 nanometers. And the reason for that is because there's stuff sticking off of it. And it's associated, so it's still considered part of the membrane, but there's stuff sticking off it. So that stuff is going to be proteins, it could be sugars, glycolipids, it could be glycoproteins, okay? It could be all sorts of things. So all of this other stuff is going to stick out off the membrane and sometimes stick through the membrane, and that is going to make this much more complex. This is a more realistic representation of what the membrane looks like than our sort of standard lipid bilayer that doesn't show any of this stuff. So, membrane proteins, there are 3 broad classes, and then there are a bunch of subcategories of the integral membrane proteins. So I'm going to call these classes to help sort of clarify this, and then the subcategories we'll call types. So 3 broad things is A, the protein can be entirely, or at least it can go somewhat into the membrane. This sort of a protein is called an integral membrane protein. So integral means it's basically integrated into there. So it doesn't actually have to span the entire thing, but it does have to have at least a significant portion stuck into the membrane. And one way we can determine that something is integral is that to get it out of the membrane, we really have to put detergent in there to break up the membrane and also then to bind to the integral membrane protein, because all this section right along here, that's all going to be very hydrophobic. So integral membrane proteins aren't going to want to fold in water, so you can't just like get it out. And then we have peripheral proteins, which are associated with the membrane, sometimes because they bind to another protein, or they could be something that is just bound directly to the membrane, like so. But these you can get off simply by maybe changing the pH or adding some salts or something like that. You don't have to add detergent to get it to go away from the membrane. So peripheral proteins, they are associated, but they do not stick into it. And then we have amphitropic proteins, and remember, amphi means both, and these are proteins that can be associated with it, and then even in the normal course of life, certain regulations or things can cause it to come off. So it has purposes where it is bound and also where it is in the liquid phase. All right. Some of these proteins also may actually be lipoproteins, where they are connected to an actual lipid. We'll talk about those. Those would be a type of the integral protein, although you could possibly get just the protein off by cleaving it. All right, so then we have subcategories of the integral membrane proteins. And I want to clarify, just point out right here, there is a typo in your book. The description, I think, the legend, it describes it properly, but the actual figure, it's wrong. So it's got type 5 and type 4 flip-flopped. And it makes sense. They start with type 1 over here, but then they just haven't flip-flopped. So just be aware that that's wrong. All right, so type 1 and 2 are going to be integral membrane proteins that have exactly one stretch of the protein that goes into the membrane. Usually what this is one alpha helix that goes through the membrane. So type 1 and type 2 are going to be, for some reason we decided to separate these into two types, but type 1 and type 2, it matters where the N-terminus of the protein is. Right? So if the N-terminus is on the outside of the cell, it is going to be a type 1. The N-terminus is on the inside, is going to be a type 2. Right? So here's inside and outside. If this was a membrane like a mitochondrial membrane or something like that, or a nuclear membrane, instead of the outside of the cell, we would say the outside of the organelle. But you can generally still define an inside and an outside. Right? So only for these two types, though, does it matter where the N-terminus is. For the other four, doesn't matter. It can be anywhere. Type 3 is probably the most common type, and this is a type where we have one chain of the protein, and it goes through multiple times. So at least 2 times in order to not be type 2 or 1 or 2, but often it's even more than that. So what you can see is it sort of goes, if this was the N-terminus, which it's not indicated, so I can't tell, but it sort of goes through. It may also have significant portions out here that are not in the membrane. Sometimes it's just loops like this look. Sometimes it might be an entire domain that can fold into its own thing. Type 4 is kind of like type 3, except look, they're different chains. So type 4 occurs when you have really this quaternary structure, bringing all these alpha helices together, versus tertiary. It doesn't have to be alpha helices. It usually is, which is why they're showing you that, but it doesn't have to be. And then type 5 and 6 are lipid anchors. So type 5 is a protein that is anchored just by a lipid. So the protein itself does not go into the membrane, but the lipid part does. And then a type 6 is one that has a lipid anchor and at least some portion of the membrane goes through. Or sorry, the protein goes through into the membrane. This is only showing you one, but again, it could be more than one. The key part here is one part that's lipid anchored and one part that's the protein in the membrane. So I like to think of this really as 3 pairs. So there's the first pair of type 1 and 2, where it's just one going through, and we have to know where the N-terminus is. And then 3 or 4, where it's multiple sections going through for sure, and we have to know whether it's one chain or multiple chains. And then 5 or 6, which is where we know there's lipid anchors, but we need to know also, does part of the protein go through? So it's really hard to get the structure of integral membrane proteins, especially because our ways of getting structures generally require the protein to not be bound to a membrane. So if you have a membrane or even detergent in there, it can be problematic to get an NMR, and it also is almost certainly not going to crystallize. So we actually still know less about membrane protein structures than we do about soluble proteins. This is changing. They are getting better at finding these structures, but it is still a lot harder to do. So in some cases, we don't know as much about them. What we do know is that a lot of transmembrane segments are alpha helical, and we can actually tell this even without getting the structure, or we can at least have some evidence for it, I would say. Maybe you can't be sure unless you get structure, but we have some evidence for it. And that's through something called a hydropathy index. So all the hydropathy index it does is it takes the amino acid sequence and then it plots the polarity of each amino acid. And what you get is sometimes you get sections where pretty much all of your amino acids are going to be nonpolar. So nonpolar is as above, polar is below. And then you'll get other sections where you have polar amino acids. And we know that this really should not be going through the membrane because the membrane is very nonpolar. And if you have you might have a few amino acids here and there. Like actually, if you look at this protein, the the amino acids sticking in this case might actually occasionally be polar and interact with each other. But you probably don't have an entire stretch of a bunch of them, because then some of those would have to be sticking into the membrane. So what we can do is we can look at how long these sections are and figure out how many amino acids it took to go through the membrane, because we assume it starts going through the membrane here and comes out on the other side here. And so it turns out that because of the width of the membrane and we know for sure how long an amino acid, or sorry, alpha helix with a certain number of amino acids is going to be, it takes about 20 to 25 amino acids to form a helix long enough to go through a membrane. So if we have, so this one, this is Bacteriorhodopsin. It's just a type, this is going to be a type 3 integral membrane protein, because we have, you see one amino terminus, it goes through a whole bunch of times, and then one carboxy terminus. We can also tell this over here from the hydropathy index, because we started numbering at, well, they're starting showing us at 10. So we started at something must have happened. Maybe we cleave off 1 through 9 or something. We start numbering at 10 and we don't restart the numbering. So if it was something with multiple strands, we would not continue the amino acid number. We'd start over at 1. So what we can see is that each of these little peak things is long enough to go through a membrane. Now it can be a little longer because maybe the alpha helix continues out here a little bit, or maybe it's sort of like this purple one, it kind of angles, so it's going to take a little longer that way. But we know they generally need to be at least that long. And we can see that all 7 of these are really long enough for that to work. So if we see a hydropathy index, you don't need the structure of a protein to do this, you just need the sequence, and that's easy. We can always get the sequence because we know the gene that it comes from, and we can sequence that. And so we can look at this and say, there look like there are 7 alpha helices that go through the membrane in this protein. So even if we didn't have the structure, for Bacteriorhodopsin we do, but even if we didn't have the structure to know exactly where they are, you could identify that that was what was happening. A protein that might be type 1 or 2 is going to be very different. You might have mostly polar sections, or like here, we have a little bit of a section that's nonpolar, but that's not nearly enough to go through the membrane. So we can tell that was not a stretch where it went through the membrane. That's probably just some portion where maybe it's folding back on itself. Well, it's about 50, so yeah, so it's over here, right? So maybe it's sort of folding back on itself, and these guys can just be pointing into the rest of the protein versus pointing into the membrane. All right, and so we see just one stretch that's really long enough to go through the membrane. In this case, this is glycophorin, which is just a different protein. In this case, we also know that it kind of loops in a little bit and then it comes back and then does the alpha helix, and part of that's going to show up in that yellow thing. But so we can tell a lot. Now, we cannot tell just by looking at this hydropathy index whether it's type 1 or 2. You would have to know where it started. So we can see that the amino terminus is on the outside of the cell, right? So if I told you that, I could say the amino terminus is, we could say here's the outside, right? And the other thing you could do is you could then determine, I wouldn't have to tell you that this is on the inside. You could say, well, if this is on the outside, so is all of this. It can't possibly have gone through yet. So then it goes through from the outside to the inside, so that you know this must be on the inside. And since the amino terminus is on the outside, this guy is a type 1. So we can have stretches that go through that are beta strands. Most of the time when we see this, we end up having a beta barrel. And these tend to actually be porins, where they're transport things and they let something go through that beta barrel. So remember, beta strands, we basically have one face that could be pointing into the membrane and then one face pointing inside the beta barrel. So that those amino acids could actually be polar, and then we could see things come through. Now, if they're polar, that's actually going to be a lot harder to identify that this is going through the membrane, because if you actually have alternating polar, nonpolar, it's not going to be as obvious. But in some cases, you can see pretty long stretches of just nonpolar amino acids as well. So let's just come back briefly to these type 5 and 6. Again, they are lipid anchored, and what that means is they are lipoproteins. They have to have an attachment somewhere between the lipid and the protein. Usually that's going to be between a side chain. It's going to be a covalent bond between a side chain and the lipid. So you can see here, this is just actually a fatty acid that has been attached to the sulfur of a cysteine side chain. This is just a thioester. We can see here that this is again a fatty acid, but this time we got attached to the N-terminus of the protein. This is the N-terminus, and it's like that N-terminal protein amino acid is a glycine residue, which is why you see that CH2. And here's a more lipid. This is one of those other lipids that was in that class. So there are these lipids like farnesyl or geranyl. These are isoprenes. They're isoprene-derived molecules. They're very fatty, so they're still considered lipids. They're not our glycerophospholipids or our sphingolipids or our cholesterol, right? So they wouldn't be sort of we wouldn't draw them like this, but they are still lipids and they can still get stuck into the membrane. And in this case, that's attached via the cysteine of a cysteine side chain. We also even can have everything involved. So this guy has, here's the lipid. We can see this is actually a glycerophospholipid. It's kind of hard to see it the way it's drawn. But here is the glycerol. It's carbon 1, carbon 2, carbon 3, right? Carbon 1, 2, 3, and there's 1, 2, 3 OHs, and there's the phosphate. But then this glycerophospholipid also has an inositol on it. That's fine, we saw some of those. But then instead of stopping there, we put more sugars on it, and eventually we link it back around to an entire protein. So this molecule has a lipid, an oligosaccharide, and a protein. It's got really all of our things except for a DNA, which we wouldn't have. All right, so all of these are going to be lipid anchored. Now, all of the ones that are shown here are going to be a type 5. Now, if this guy sort of came around and stuck back in, that would be a type 6. I've mentioned before that membranes are not static the way we draw them. They are moving. Pretty much all molecules are moving unless they're in a very, very cold crystal. So they're moving, and we can think about sort of two idealized states. One is the liquid ordered state, and this is almost really what we think of, what we've been drawing with our little head groups, and then the fatty acids just stick straight down, and you kind of think of them as packing all next to each other perfectly. And then the other extreme is the liquid disordered state, which is essentially almost like tangled up spaghetti where everything's pointing every which way and crazy, right? In reality, all membranes are going to be somewhere in between these two, and where they lie on the continuum between liquid ordered and liquid disordered is going to depend on the temperature as well as the type of fatty acids. So the longer things tend to be more liquid ordered, and shorter tend to be more liquid disordered. Also, more saturated things tend to pack more in the lipid ordered. This makes perfect sense because remember, these are the ones that kind of just sort of go in a line. And more unsaturated, with those kinks from the double bonds, tend to pack more into the liquid disordered state. So if you have a bunch of really long saturated fatty acids, you're very likely to be pretty close to liquid order. If you have a bunch of short unsaturated ones, you'll be liquid disordered. If you have like short saturated or long unsaturated or a mix, you'll be kind of in between the two. Another thing that can play a role is cholesterol, and what cholesterol does actually is it does not do the same thing to all membranes. What it does is it shifts it towards whatever those lipids would not naturally be. So if you have long saturated things and you put in more cholesterol, it actually pushes it more towards the liquid disordered state. You can think about that because try to imagine now shoving a cholesterol into this nice ordered state. It's going to disrupt some stuff. On the other hand, if you put cholesterol into something that's very liquid disordered, it's actually going to make it more ordered. And again, this is because if you think about shoving a cholesterol in, it sort of has to now be a little bit more lined up, at least around the cholesterol, because the cholesterol is really pretty set. It's not going to be, it can't flop around like that. It's all those rings. And then the other thing that makes a big difference is temperature. So the colder it is, the more it's going to be in the liquid ordered state. The hotter it is, the more it's going to be in the liquid disordered state. So for human membranes, this is not a particularly important thing because our membranes, like our rest of our bodies, are going to be in a pretty narrow range of temperatures, right? Maybe between, say, 95 if you're really starting to get hypothermic, up to maybe 104 if you have a really bad fever. So maybe 10 degrees we've got, right? Before you got bigger problems. Something like bacteria or plants, however, that have to withstand much more different temperatures, right? I mean, plants around in the upstate New York area might have to withstand a temperature range of maybe 100 degrees. 100 degrees Fahrenheit. So I was talking Fahrenheit the whole time, so questionable, but 100 degrees Fahrenheit compared to, say, 10 degrees Fahrenheit that humans do, right? Because maybe in the really coldest of the winter, it's maybe 0. In the really hottest, maybe it's getting close up to 100. So that's going to be a big, big difference. And this is one of the reasons that plants and bacteria tend to have many more different types of fatty acids that they can make. So bacteria cells use this ability to swap in and out their lipids to maintain a constant fluidity, a constant structure between those liquid ordered and liquid disordered states, even in a wide range of temperatures. So what we see is that at low temperatures, where it has a tendency to be more more liquid ordered, we will see fatty acids that tend to make it more disordered. So we'll see short things and we will see unsaturated things, right? So we see a lot more unsaturated things at this cold temperature compared to the warm temperature where we see almost all saturated fatty acids. Because at the warm temperature, it's actually trying to make it more ordered. So it's going to put in longer things and more saturated things. So let's talk about the individual molecules. Again, these are not sitting still in their one part of the membrane. They are moving around. So talk about two different types of movement. One is lateral diffusion, which basically means moving within the same layer of the bilayer. So that happens very fast. So you can have one molecule, if you could track it, moves over here. But it's still on this, let's say this is the outside. It's not labeled, but we could say this still on the outside bilayer. Transbilayer diffusion, which would be taking one on, say, the outside and moving it to the inside bilayer, that is very, very slow. Days instead of micrometers per second that it can move, right? So that's a very big difference. And the reason for that is simple. It's that to move from one bilayer to the other, you have to have an intermediate bit in here where you have your polar head group smashed right in the middle of that lipid bilayer in the most non-polar section of it. So that's obviously not ideal. That's almost like the a very high activation energy of a reaction. It's not a reaction, but you could think about it like that, right? You have to put it into a very high energy state to get it to then go back down to the good energy state where it's on the other side. So that's going to happen very slowly on its own. Okay, if you want this to happen, you're going to need to use a protein to help it do that. So there are a number of proteins that can do this, and actually the simplest one is scramblase. So what scramblase does is essentially all it has is it's got a section here where it will allow that polar head group to be there. It's got some polar amino acids and such. So it's just essentially just letting the lipid move on its own. Okay. However, it was moving toward equilibrium. So what that means is if there's more of this lipid on the outside than the inside, if scramblase is around, this lipid will move preferentially towards the inside. If there's more of it on the inside than the outside, it'll move preferentially this way. And scramblase is not a particularly specific enzyme. It just kind of lets most lipids go through. Flippase and floppase are more specific, and these guys are going to catalyze the movement of sometimes even pretty specific lipids. So flippase only moves phosphatidylethanolamine and phosphatidylserine, and not the other ones. Floppase is a little bit more non-specific. It just moves any phospholipid. But what these do is they move things to where they actually are already higher concentration. So this is not energetically favorable. We're going to talk more about that in the next lecture, but it's not energetically favorable. Here's sort of an introduction for you. So we can actually see something that requires cleaving ATP to get the energy. So this might move, this usually only moves it in one direction. So flippase moves it from the outer to the inner. Floppase is going to move it from the inner to the outer. And so this would only move, this can move phosphatidylethanolamine. Even if there's already more phosphatidylethanolamine on the inside, it can move it from the outside to the inside. Floppase being a little bit more nonspecific, you might imagine, you could see why you might need that, right? So all of the lipids that you're going to make for your lipid bilayer, they're going to be made inside the cell. You have to get them outside somehow, right? You're not just pulling these in randomly from the outside. So you have to get them outside, and if we're going to have, remember those bilayers did have different concentrations of different lipids, and so the ones that are higher in the outer bilayer, you're going to have to move them to the more concentrated side somehow, right? And so it makes sense why we would need, for sure, why we would need something like floppase. Floppase also requires ATP to do that. So a lot of the times I tell you things and you essentially just have to believe me, and I'm not going to tell you how we learned this stuff. I try to avoid that when possible, but a lot of times we do. And this is just one way to talk about how we know about that movement. And FRAP is really good for actually that movement within the bilayer. So FRAP stands for fluorescence recovery after photobleaching. So a little bit of background here. When you have fluorescent molecules, so fluorescent molecules are going to absorb, it's usually UV light, and then give it back off. If you stick an intense light at them, so like an intense laser beam, it will bleach out the molecules so they can't do that anymore. And this is a known property of fluorescent things. So what you can do is you can put fluorescent probes on, say, all the lipids in the cell. And then when you look at it, the cell is glowing red or whatever. And you can look at it very, very zoomed in and just one little patch here. Then you can shoot the laser beam at just that little patch, and what you'll see is a little circle where it's not red. And I want to be clear, the lipids are still there. This is not a hole. We did not poke a hole. What we did is we just, we photo-bleached the fluorescent probes so we can't see the red anymore, but the lipids are still there. And then you simply watch with a fluorescent microscope to see how long it takes before you see fluorescence in that hole again. And since we know that the ones that were there, they can't recover from this. They cannot start absorbing fluorescing again. What that means is lipids that were not in that little circle have moved in. And presumably the ones that were in the circle have moved out to make space for them. And so you can watch that rate. And this is how we know that it's actually very fast for this to move in. We actually can now watch, we have ways of watching a single molecule in fluorescent microscopes, and so you can also do that. You can label, say, one molecule and then just literally watch it. It's pretty crazy how good we are at things like this now. And what we see actually is there is movement, but it is still somewhat constrained. It's almost like there are little patches. So let's say this is one patch, and you start it here, and it moves all around, but it seems to be staying in this general vicinity. And then maybe it ends up over here, and once it ends up over here, it doesn't go back here. And now it stays in this general vicinity for a while, and it moves around, and eventually it goes over here, and then once it's there, it's stuck, and it goes over here, and then eventually it's going to go over here. I like to think of this, I have one of those vacuums that, like the robot vacuums. It's not a very good one, and so what it does a lot is it bounces around one room for a while until it accidentally hits the door, and then it goes into the next room. Once it's in the next room, it's usually stuck there, or it doesn't go back into the first room for a while, and that's sort of how this is working, right? So it's kind of, there's clearly some point at which it can move across, but then it's sort of constrained within there. And one reason is probably because this is, again, the monolayers is not as simple as we think it is. There are proteins in there. There might be things even on the inside of the cell. There might be actin molecules sort of bound along, keeping the cell structured. There might be all sorts of stuff going on. And so where these proteins are might be what's happening. So you might have that the lipids bouncing around because it keeps bumping into proteins, and only when it gets to a spot where there's enough space to really easily go between them, right, then it can move. And the proteins can move, but they generally don't move super far, super fast, right? So they have, again, some constraints here. It's because, again, everything's more complicated, and then we're going to simplify it, but there's a lot going on. So there even are little parts of the membrane that are called rafts, and these are just microdomains, which simply means that this portion of that lipid bilayer is different from the rest of it. And it can be a pretty significant portion of it, so lipid rafts can sometimes make up 30, 40, 50% of the membrane. So they're not necessarily just a small portion, but so you might have like this membrane, and you have like a lipid raft here, another lipid raft here, right? So they can be kind of a big chunk of it. And it really just means it's made of other things. So the lipid rafts are often more enriched in cholesterol. They may be more enriched in sphingolipids than the rest of the lipid bilayer. They may have certain different proteins that are more likely to be there. And what these are is they're just areas where certain things are going to happen. So I don't want us to worry too much about the details there, but sometimes there are sections where maybe there's a signaling pathway that it's all happening at that particular lipid raft, or maybe there's a section that is actually where it allows stuff to come in and make little endosomes. Something that is sometimes considered a lipid raft and sometimes not are these things called caveoli. And caveoli are areas of the plasma membrane that, so they start out like so, and they can basically invert in so much that eventually they pinch off, and then we have a vesicle on the inside of the cell. And the reason they're called caveoli is because there is a protein called caveolin that this is an example of one of our type 6 integral membrane proteins, right? Because we see here it's got lipid anchors, and it also has a stretch of the protein that goes in through. And it also associates with cholesterol, so that's, a whole separate thing. But so caveolin is the way caveolin inserts itself into the inner bilayer, or the inner part of the bilayer. It actually causes it to bend the other direction, right? So normally we would think that the plasma bilayer is kind of, bending out a little bit in most places. The caveola causes it to bend in, and eventually it can bend into the point, like I said, of making this little vesicle. So this can be used to bring in certain things sort of wholesale from the outside of the cell. So we're going to talk next time about how we have certain transporters to let very specific molecules in. But if you kind of just want to bring in a section of the outer liquid, caveola can do that. This can also be a way to almost eat the stuff on the outside. So you can bring in molecules and things that are out there, and then they're in these vesicles, and then at that point other systems can trigger maybe them coming out of the vesicles or being brought to different parts of the cell or whatnot. right? So I don't want us to worry too much about the lipid rafts or the caveola, except that I really do want you to recognize that the membranes are not uniform. They are not just these little lipid layers, right? That they're much more complicated than that, and they have a lot more diversity and variety. Right, so that is our introduction to our membranes.